Okay, good afternoon. Everybody can hear me? Good? Okay, my name is Vikram Gadagkar, and I'm an assistant professor at the Zuckerman Mind-Brain Behavior Institute at Columbia University. It's a great pleasure to be here today, and thank you so much, Shiri, for inviting me. Since I'm visiting from New York, I thought I would begin with a New York story. The great pianist Arthur Rubinstein was walking along the streets of New York one day when a pedestrian approached him and asked him, "How do I get to Carnegie Hall?" And Rubinstein pondered this for a second and said, "Practice." So indeed, many of our motor skills are not innately programmed, but are acquired through a process of trial and error or practice. The songbird is an excellent model to study how motor skills are learned through trial and error. The adult zebrafinch song is a naturally learned, highly stereotyped motor sequence. By looking at the spectrogram of the song, you can see that the song consists of distinct syllables, A, B, C, and so on, that the bird repeats again and again and again. A juvenile zebrafinch is not born being able to sing like this. All the juvenile can do, just like a human infant, is babble. The father finch sings his song to the juvenile remembers the father's song, and then spends the next 2 or 3 months of his life learning to make a copy of the father's song through a process that looks very similar to trial and error learning or practice. Here is a video of a father finch tutoring his son. So the question that I was very interested in is, how does the zebrafinch brain evaluate its own performance during practice? A clue to the neural mechanisms underlying trial and error learning comes from the study of animals learning simple tasks for external rewards like food or juice. Put a rat in a box with a lever, and he will learn by trial and error to press the lever for a food reward. Classic experiments have shown that unexpected rewards lead to dopamine increases in the brain, and lack of expected rewards lead to dopamine decreases. These dopamine increases and decreases are thought to positively and negatively reinforce preceding actions, thus leading to learning. However, most of our motor skills, like speaking or playing a musical instrument, are not learned in the pursuit of immediate food or juice rewards, but are instead learned by comparing ongoing performance to internal goals. Is it possible that dopamine might also reflect better or worse performance relative to internal benchmarks? To answer this question, we need to look inside the songbird brain. One of the great advantages of the songbird model is that the songbird brain contains a discrete set of interconnected brain regions called the song system that is dedicated to learning and producing song. The song system includes a dopaminergic projection from the nucleus VTA to area X, the singing-related basal ganglia. These brain regions, spanning dopamine, basal ganglia, thalamus, and cortical areas, are evolutionarily conserved, and mammals also possess similar brain circuits. Based on these similarities, we and others hypothesized that these neurons in VTA might convey a conceptually similar evaluation signal for song learning, even though song is internally evaluated. When I was a postdoc with Jesse Goldberg at Cornell, I tested this hypothesis by recording from antidromically identified area X projecting VTA neurons in singing birds. These were adult birds that had already learned to sing, but I fooled them into thinking they made a mistake. To do this, I selectively distorted 50% of the renditions of a particular target syllable with auditory feedback. What do I mean by this? So let's say the bird song has 4 syllables, A, B, C, D, which sounds like chikwa, and I take the first part of syllable A, which sounds like ch, and play it over syllable D. So the bird is singing along, chikwa, 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 but what the bird hears is chikwa, 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 depending on whether or not syllable D was distorted with feedback. The idea is that a distorted syllable sounds wrong or sounds bad to the bird and should cause dopamine decreases. Just as we had predicted, we found that these dopamine neurons were suppressed immediately after distorted syllables. We also found that these same neurons were activated after undistorted renditions of that same syllable. Our interpretation is that because we were distorting syllable D, for example, half the time, the expected quality of syllable D was lowered for the bird. So when the bird heard an undistorted syllable D, even though it was completely untouched, it actually sounded better than expected, and that is what is signaled by this phasic activation. So here we have a better than expected and worse than expected dopamine signal, not for external rewards like food or juice, but for the bird's own performance. That is, whether its performance was better or worse than it expected it to be. We were very excited about these results because they identified the long sought after evaluation signal for song learning. And more broadly, they suggested that evaluation might be a general principle of the brain's reward system, including the internal evaluation of complex motor sequences. I was telling some of my musician friends about these results, and they claimed that they treat mistakes very differently during practice and performance. During practice, they pay a lot of attention to their mistakes because they want to learn from them. But if they're performing and it's the big day and they're at Carnegie Hall and then they happen to make a similar mistake, they might brush it under the carpet because, well, the show must go on. This led us to wonder, can the brain actually handle motor errors differently during practice and performance? It turns out that zebra finches are once again a great system to ask this question because male zebra finches practice their songs alone with the ultimate goal of performing to a female. It's a courtship song. But to answer these and similar questions, it turns out that the method of electrophysiology that I had been using is not well suited because we could record neurons only for minutes at a time. To solve this problem, we developed the first dopamine photometry system in singing birds. In dopamine photometry, we use a virus to inject a genetically encoded dopamine receptor into area X. When dopamine binds to these special receptors, they release light, which can be picked up with an optical fiber. The amount of light now is a proxy for the amount of dopamine in that region. Dopamine photometry has been a real game changer, as experiments that would literally take years with electrophysiology can now be performed in a matter of weeks. The result I'm going to show you next was done in collaboration with Andrea Roser, a talented grad student in Jesse's lab. But before that, here's a video of a male who sees a female and immediately starts singing female-directed song to her. Where did you go? Okay, so the question now becomes, what happens to this dopaminergic evaluation signal that was present during practice during female-directed performance? So just as my musician friends might have predicted, we found that the dopaminergic evaluation signal that was present during practice was substantially reduced during female-directed performance, suggesting that the brain self-evaluation system is active during practice but turned down during performance. We then wondered that maybe you don't care so much about your own mistakes when you're performing, but perhaps you care a lot about how the audience is responding to you. In this case, the audience is the female. Female zebra finches don't sing, but they make these short vocalizations called calls. They're always going like And in fact, we found that the dopamine system was robustly activated by female calls, especially when the calls overlapped with male song. These results are the first demonstration of the social modulation of dopaminergic evaluation signals. And more broadly, they show that dopamine neurons can change their tuning from self-evaluation during practice to social feedback during performance. The discovery of dopaminergic performance evaluation signals in adults sets the stage for us to finally ask if dopamine guides vocal learning during development. My student John Caston recently performed the dream experiment of continuously monitoring dopamine for weeks in juveniles as they learned to sing. This work was done in collaboration with Alison Duffy, Adrian Fairhall, and Kim Stackenfeld. We recorded every single vocalization produced by a juvenile between day 61 to 100 days of age, which spans what we call the plastic song phase of learning, where syllables can be identified but are still being refined, and into early adulthood. We simultaneously recorded every single dopamine transient associated with these vocalizations. Now that we're analyzing natural song with no experimentally induced perturbations, we need to have a way of defining song quality. That is, what is a high-quality syllable and what is a low-quality syllable? We first parameterize every rendition of a given syllable into a set of acoustic features like pitch, and spectral entropy, amplitude, and so on, allowing us to place these renditions into a multidimensional acoustic feature space. In this cartoon, each circle represents the rendition of a juvenile syllable, with T denoting the current rendition, T minus 1 the previous rendition, and so on. These juvenile renditions can be placed into this acoustic feature space as shown below. Now we can also place the adult version of this syllable, denoted by the black square, on this same feature space. Then for any given juvenile rendition, we can ask, is it closer to or farther from the adult version compared to recent renditions? If it is closer to the adult version, we say it's a high quality rendition. And if it is farther away from the adult version, we say it's a low quality rendition. Then the question becomes, is high syllable quality associated with high dopamine? Here's an example syllable from our data set with the spectrograms aligned to a closer high quality rendition on top and a farther away low quality rendition below. John took all the renditions of this syllable during the course of a day, and he picked the top 10% of the renditions that were the highest quality, that were closest to the adult version. And he plotted the corresponding dopamine responses on this heat map. Here, each row is the average dopamine response for the top 10% highest quality renditions on a given day, going from day 61 to a day 100. Similarly, he picked the bottom 10% lowest quality renditions, or ones that were farthest away from the adult version, and plotted their dopamine responses on this heat map. The blue trace here is the average dopamine response across development for the high quality renditions, and the red trace similarly is the average dopamine response for the bottom 10% lowest quality renditions across development. So you can see from these plots that, as we expected, high quality renditions are associated with high dopamine and low quality renditions with low dopamine. We analyzed 25 syllables across 6 birds in our data set, and on these heat maps, we show the average dopamine response across development averaged for that syllable on each row, giving us 25 rows for 25 syllables. We found that for each of the syllables we looked at, the dopamine response on the high quality renditions was significantly more than the dopamine for the low quality renditions. So far what I've told you is that dopamine is correlated with syllable quality, right? High quality renditions are associated with high dopamine and low quality renditions with low dopamine. Next, we wanted to ask if we can directly test if dopamine guides song learning. That is, does dopamine care about how the song moves in the future, and does it actually guide this movement? In this cartoon, each circle represents the rendition of a given syllable during the course of a day. And O denotes the renditions early in the day, and X denotes the renditions later in the day. Now suppose you look at syllable renditions early in the day, and you find that high-pitch renditions have high dopamine associated with them, and low-pitch renditions have low dopamine associated with them. If you found this, then you would expect the pitch of the syllable to increase during the course of a day, because dopamine is positively reinforcing. This is exactly the kind of thing we looked for, except not just for pitch, but for all the features in our data set. In this plot, each circle represents the location of a juvenile syllable early in the day, with the color denoting the amount of dopamine associated with them. Yellow renditions had more dopamine associated with them, and blue renditions had less dopamine associated with them. Now, in this feature space, we can construct a vector called the dopamine vector, denoted by the red arrow, which goes from low dopamine to high dopamine. This is the direction in feature space that the song should move if dopamine was guiding song learning. The gray X's denote the locations of the renditions later in the day, with the black arrow denoting the direction of song movement, going from the black circle, the median location of renditions early in the day, to the black X, the median location of renditions later in the day. Now, just looking at this example, you can see that song does seem to have moved in the direction predicted by dopamine. Now, of course, this is just one example syllable and a single X block on one day. We can consider all possible future X blocks and average our data across all the days and all the syllables in our data set to give us a combined plot that looks like this. The X-axis is renditions in the future, and the Y-axis is movement along the dopamine vector. The blue line with light blue shading is the mean and standard error of the real data, while the black line with gray shading represents shuffled data, where we shuffle the correspondence between the syllable renditions and the dopamine transients. What this tells you is that song seems to move significantly more in the direction predicted by dopamine for the real data compared to shuffled controls, suggesting that dopamine is actually guiding vocal learning. Yeah?
Just to check, so the previous slide was your grad student rating the bird's song, and that notion of learning checked out. This slide is talking about the bird's own evaluation of its syllables, and this is also saying that this notion of learning checks out, yeah?
No, they're both using the same metric of how close the song is to the adult version.
But this is measured in terms of the bird's dopamine. Previous one was measured in terms of your grad student's estimate of the top 10 or bottom 10% of syllables.
No, I would say that the metric is the same. It's just that this is a more sophisticated analysis looking at like the evolution of the song and the direction of dopamine. And the previous slide was a much more coarse-grained analysis of just looking at bottom versus 10 with very little sort of temporal information. Yeah. Okay. Finally, we wanted to, so I've been telling you so far that dopamine encodes a performance prediction error. That is, dopamine cares about the quality of the syllable relative to previous renditions. Next, we wanted to ask if this is actually the case during development. So the question is, does dopamine encode a performance prediction error where it cares about the quality of a syllable rendition relative to what happened before, or does it simply encode a mismatch error where it only cares about whether a given syllable is good or bad independent of what happened before? So here is the reinforcement learning model simulation for a simple mismatch error. The x-axis is rendition relative to dopamine response. So 0 is the current rendition, -1 the previous rendition, and so on. And the y-axis is regression coefficient on error. All that means is that is the correlation of dopamine on the current trial with syllable quality. So for a simple mismatch error, dopamine on the current rendition is positively correlated with the quality of the current rendition, but uncorrelated with the quality of previous renditions, because all it cares about is how you did now. However, in a performance prediction error model, dopamine is once again positively correlated with the quality of the current rendition, but negatively correlated with the quality of previous renditions, because the worse those renditions are, the better the current rendition will sound in comparison. We found that our data much more closely match the performance prediction error model, showing that dopamine does seem to encode a performance prediction error during development. Taken together, these results show that natural behavior can indeed be learned through dopamine-mediated reinforcement. I would now like to take 2 minutes to talk about the big questions that we're trying to tackle in the lab. We are broadly interested in the neural mechanisms of performance evaluation. So imagine, for example, that you're learning to play my favorite Beatles song, Yellow Submarine. You first memorize the song as a memory trace in your brain. You then practice the song through self-evaluation before finally performing the song to your friends, who might then evaluate your performance. Where these memories are stored, how we evaluate our own actions during practice, and how we evaluate the actions of others are major unsolved questions in neuroscience. The songbird is an excellent model to address these questions, because juvenile male songbirds first memorize their father's songs. They then learn to produce the song through practice, with the ultimate goal of performing to a female, and females need to evaluate the quality of male song. The big questions we are trying to answer in the lab are, first, where in the brain is this song memory, or the so-called template of the father's song? Second, now that we have identified the dopaminergic evaluation signal, what are the causes and consequences of this signal? In other words, how do upstream circuits actually construct this evaluation signal, and how do downstream circuits use this evaluation signal to learn song? And finally, we are studying female songbirds to investigate how we evaluate the actions of others. I thank all the amazing members of my lab, my funding sources, And I thank you for your attention. Yeah.
Oh, okay.
Okay, go ahead.
So my question maybe is a little not directly related to specifically practice versus performance, but it's about birdsong in general.
Yeah.
So one question is it possible for a baby bird to learn a different song?
Yes. So, yeah, within limits, right? So you try to teach a baby bird, Michael Jackson, it might not work.
Right.
But if you try to teach it like a different zebra finches song, or even a song of a species that's like closely related,
Okay.
they can do that.
And okay, so I guess one question is are you aware of work that looks at like how or if the song has changed over generations?
There is interesting work looking. Actually, this reminds me. So, my favorite experiment of all time, I think I'm going to tell you about that. Okay, so there's this guy called Ofer Chernikovsky who's at Hunter College in New York, and he did this really cool experiment. So what he did was, it's sort of related to what you're asking. He took a baby bird, and he said, what happens if I take away the father, right? If there's no father, there's no model for the baby to imitate, what happens? So if you do that, these are called isolate birds, they end up singing, what might be described as a crappy song. Okay, it doesn't sound zebra finch-like, it sounds screechy, it just sounds bad, right? That's what they end up singing. And if you show a female to this bird, he doesn't, that's all he's got, he's going to sing this crappy song to the female. And if the female doesn't have a choice, she's going to mate with him, and they're going to have babies. And he's going to sing this crappy song to his son. And if the son doesn't have a choice, he's going to try to make a copy of this crappy song, right? But now, if you do this for 5 generations, the song is back. The song is back to sounding like a zebra finch song. It sounds perfectly normal. It doesn't sound screechy, doesn't sound bad. So the idea is that there is a small innate predisposition in these birds to make species-typical sounds. And even if that force is like 5%, you can imagine it compounding from generation to generation, and the song is back. And there are very interesting parallels in human language where this also happens. So yeah, but I will stop there. Yes, actually you had a question at the back first. Go ahead, you had your hand raised.
No, just a continuation of
Sure,
the experiment. In that case, if you do it twice on 2 separate specimens, will you get the same song eventually, or so repeating the five generations of starting from the crappy song and then converging? Will they both converge on the same kind of?
No, they will not. They will not converge on the same
so
song.
It's like a manifold of songs.
Exactly. They will converge. It will be indistinguishable from the cloud.
Interesting.
Yeah. Go ahead.
So yeah, amygdala. So in songbirds we don't have mammalian style amygdala, and we know in humans it's play a significant role in these dopamine systems and the impact of them in the dopamine reward systems. So how do you, if you want to speculate about human performance as you bring these human images, how do you want to tweak your models when you don't have like that mammalian style amygdala in your system to be able to like have a right estimation on human vocal learning systems when we have amygdala?
So the birds also have an amygdala, but why do you need the amygdala specifically?
So because they are like center for all of these socials and all of these centers next to the amygdala who release the dopamine. Then you have all of these social interactions. They definitely impact.
I mean, the amygdala definitely impacts it, but
it's not in your model. So
yeah, I mean
the songbird's model of vocal learning, you don't have amygdala, of course.
So that's not strictly true, is what I'm saying. So people like Jesse Goldberg are now looking at like inputs to dopamine through these sort of traditionally non-song system areas like the habenula, like the habenula, amygdala, and things like that. And they are finding that actually these areas do contribute to song learning. That's one. And the second thing I will say is that there's actually been some very interesting experiments in humans which look at performance prediction errors. So one of the experiments is they took piano players and they had EEG being recorded in the piano players while they were playing a piano. And they also had headphones so they could listen to what they were playing. And every once in a while, they introduced errors into their playing. So very similar to what we did for the birds earlier. And they also found very similar prediction error signals called error-related negativity, which in the anterior cingulate cortex, which has a massive dopaminergic projection. And so we think that very similar things are going on in mammals and humans as well during performance. Gilly?
Do you know, is there a like critical period from the time that the juvenile heard the father's song in which it needs to start?
Yeah, so there are actually two critical periods. So there's one critical period for sensory learning. So there's a critical period for acquisition of the template. And there's a second critical period for sensory motor learning, which is the practice. And these two critical periods overlap in zebra finches. But there are two distinct critical periods.
So like, do you know like how long, so, okay, so there's like critical period for development, but also is there like a time that memory dies out? You know, like.
You mean after they reach adulthood?
No, before they, when they're juveniles, like I hear the dad's song, but I haven't practiced, and like how long can the memory last?
The memory, I think, from what we can tell, can last forever. But the critical period is for them to learn to produce the song. And that's a whole other question of how this memory lasts forever, and that we're very, very interested in those questions as well. Yeah.
Maybe a follow-up question. So in computational generative models, there used to be a very popular method called a GAN, where you have
Called what?
GANs. And so generative adversarial networks. And so the idea was that you would have a generator that would produce something like an image or a song, and then you would have a critic that, or a discriminator that would say, okay, the produced song you produced, is it good or is it bad?
Right.
And this was a beautiful theory, except for the fact that in practice to optimize it was a pain because you had to schedule the two systems such as one doesn't beat the other. Because if your discriminator is too strong, which maybe you could say, you know, for humans, by the time you get to adulthood, it's really hard to learn violin because you know that you sound terrible, right, when you start it.
Right.
One of the theories
Right.
that, you know, children don't
Right.
really have that discriminator yet, and so they're very willing to explore. You know. So I'm just wondering, how, like, do you have any
Yeah, we do.
knowledge about
Yeah.
how this is organized?
Yeah, absolutely. So I think in our field, I think it's the same thing. We call it actor-critic model.
Okay.
I think it's a very similar thing. So there's an actor that produces an action, and there's a critic that evaluates, and then that information is integrated by the learning system to improve performance. So we call it the actor-critic model for reinforcement learning, and the song learning seems to map on beautifully onto this actor-critic framework. And again, Jesse and Redong Chen have a nice opinion piece making this connection. And Mark Goldman has recently been working on a paper, I think it's not out yet, where he does a much more sort of rigorous mapping of how this actor-critic framework can lead to song learning, including the rates and like what kind of coordination you need and everything. Yeah. Yes.
I'm curious about the difference between prediction error and performance error, because sometimes I think like when I'm like playing an instrument, I like know I made a mistake and then like I expect a good mistake, so hearing it isn't unexpected. In other cases, maybe I truly like just didn't even realize that I did the wrong thing, and I'm curious if there would be like a way to distinguish between these two cases.
Yeah, that's a great question. So in the birds, I'm not sure. Again, in the birds, you know, we can tweak their expectation, right? So we can make them more or less likely to think that they make a mistake, and the dopamine, the amount of dopamine they get, scales with that. So for example, so we could do that. In the human case, also you can do things like that. In fact, in the same piano experiment, they found very interesting differences between novices and experts. So when they introduced errors, when the experts were playing, they found very different neural signatures. And the idea is that the experts knew that they're not making that mistake. You know, they're like, I did not do that. And so it's weird in a different way. But the novices were more like, oh, I guess I pressed the wrong key. And so the brain interprets that very differently.
So
Any other questions? Yes.
So there's like two interventional things you could do to like really close the loop on this. Right? One is that you could, when they make a mistake, you could play the correct syllable over, ideally getting like a positive dopamine burst and reinforcing this incorrect syllable that the bird actually sung. The other one would be that you could directly drive the dopamine
Yeah.
and cause them to learn a totally arbitrary song.
So this has been done. Yeah.
Okay,
So
so-
you can use optogenetics to, for example, activate dopamine neurons
Yeah.
only when they sing high-pitch renditions, for example, and that will drive them to move the pitch up. So you can do these kind of causal experiments.
And this is like,
It's been done.
you see exactly what you
Yeah.
hoped. Okay.
Yeah. Yes.
I guess what is it that you were referring to human analogs to?
Oh, so yeah, so there are these pigeon languages, so when people migrate from one region to another, they often speak the new language without grammar or without proper grammar, and they speak this to their children, and the children can inject grammar de novo into these languages without ever having heard correct grammar from outside. And so that's the parallel I was
Yeah,
talking.
I guess what I'm surprised by for the birds in particular is that the actual song itself that's specific for the art.
There is no actual song, right?
Oh, okay, well let's
Yeah,
talk.
yeah, that's I guess related to the question there. That the song that emerges is not like a particular song, but it's indistinguishable from the cloud of zebrafinch songs.
Okay.
Okay.
True also for the juvenile that's learning from the father is not going to mimic the exact song, but just going to be in the same distribution.
No, when the juvenile is actually learning from the father, he makes a pretty good copy of the father's song, but not identical. There is some individuality, he adds a little bit of his own twist, and that's also very interesting. In fact, when the father's song is very weird, he tends to bring the song back and make it less weird.
But I think there's stuff there that we can discuss
Yeah.
later. Okay.
Cool. Thank you very much.
Thank you so much. Thank you, Vikram. Do you need
Is it on?
Meine
Yeah, it's on. Right.
Weizmann-Institut.